Thanks for bearing with me. So I gave a version of this talk, or I guess an earlier talk on this project at last year's meeting. It was very preliminary then, and now we made a lot of progress. So those of you who were there can kind of see how things have changed. But the goal of the project that I'll be presenting on is basically just to take all the CABO data, the leaf level data, and the traits, and to use the leaf level data to see how well can we predict these traits, and then how well do those predictions sort of generalize across different settings. So in general, the way I'm thinking about things is or maybe I should say a holy grail of leaf level spectroscopy. Right? There are many things that we can do, but one of the things that we'd like to do is to be able to take spectra from a wide variety of different kinds of leaf tissue, and put these, measure the spectrum on all these leaves, and eventually put them into some kind of model that allows us to make predictions that are really general across different kinds of settings. And so, like a good example of that is Shan Kothari's paper from a couple of years ago, where he basically did this for LMA. And the title of the paper, he made sure to emphasize, is from the tropics to the tundra. It really goes all over the world, and no matter what leaf you're measuring, you can make a pretty good prediction of LMA with a single model. And we want to see, can we do this? So with a broader range of traits. So using the CABO data set, can we build trait models across functional groups, minus that first across? And how well do these models kind of generalize to new settings? It's really a very basic goal. But potentially, if we can do this, then those models will be really useful for people who want to do kind of functional ecology using spectra. It's a relatively easy thing to measure that would potentially give you a lot of information about many different traits, and so it can make certain aspects of functional ecology easier to study. So to deal with this first question, I took the CABO data from all kinds of different ecosystems across Canada, and even warrens in the Warren Kind sequence in Australia. So we had trees, we had grasses, and peatland plants and stuff like that. So there's quite a few different functional groups and different ecosystems that are represented in here. And altogether, it's around 2,000 samples, more than 100 species. Broadleaf trees really dominated, but there are quite a few other functional groups too. And this is something that I think sets the CABO data apart from many other data sets from which people have tried to do similar things. If you summarize it into all the different spectra into a single figure, you get something like this. So most of the spectra. So the middle line is the black line is the median at each wavelength, and then you have the 25 through 75 percentile, and then the light blue, which might be hard to see, is 2.5 to 97.5. So most of the spectra are within a pretty narrow range, but there are a fair number of spectra also that then are quite different from the rest. And on the previous slide, I think I also wrote that we measured transmittance spectra as well and absorptance spectra. I won't be discussing those in as much depth, but we also have models to predict traits from those. It's not as central because fewer labs actually measure or even have the tools to measure those transmittance spectra. So it's like a less, it's probably for I think the reflectance spectra are probably what most people would find most useful. And then we have this wide range of traits, and these traits represent represent different aspects of leaf function. So we have some of your classic leaf economic spectrum traits like leaf mass area, nitrogen content. There are things that relate to leaf toughness and other soluble decomposability, like some of these carbon fractions. There are pigments and many different elements, which if you look at the kind of literature on things like the leaf economic spectrum, they typically only focus on a couple different. They typically only focus on a handful of traits, and so one of the strengths of this data set also is that we can potentially get at more different aspects of leaf function. And to predict the traits from the spectra, they used a partial least squares modeling approach. I think many of you are probably familiar with PLSR as a technique. But the particular approach that I use here is based on work that like Shan Kothari and other people have done, where they divide the full, so here I divide the full data set into calibration and validation subsets. I use PLSR with cross-validation to get a number of components for each trait, and then put that number of components, even the number of components, in doing a sort of jackknife analysis, where I divide this calibration data set further into 70% and 30% subsets, again randomly, and redid that resampling over and over to get 100 different models. And then I applied that ensemble of 100 different models to the validation data set. The reason why we do this kind of jackknife analysis is just to see some, to get some sort of measure of sensitivity to having randomly, sensitivity to random variation in which data are included. But it's not like most of what I'm going to show you here doesn't really rely on understanding this jackknife analysis. And for the next few slides, I'm just going to show a representative example of how this kind of approach fares for different traits. So for example, with certain traits like leaf map per area, we do quite a good job. We can get R squared of more than 0.85 or so. The models are fairly, you might know, so the number of components is like the number of latent variables from the spectrum that are used in predicting the trait, and so it's quite a complicated model, which I think is just because there's so much data that kind of subtle aspects of the spectra can be pulled out to aid in prediction. We can do pretty well, and then for a trait like cellulose, again we do pretty well, but a lot of this needs to be driven by the fact that graminoids have considerably more cellulose than pretty much any other functional group here, and so the amount of cellulose that's in the leaves, some of this, in this case, anything that sets graminoids apart from everything else, whether it's causally, whether it's an absorption feature of cellulose or not, could be used in that model. So so it's unclear how much of it is mechanistically driven by cellulose per se. Leaf nitrogen, again, I'm showing these in kind of descending order of performance. These is kind of representative sample in descending order of performance. So things will sort of start to fall apart the further I go. So leaf nitrogen, again, it looks like a pretty sharp correlation, and again across functional group, the line within for each functional group seems to be pretty well coincident with the one-to-one line. Chlorophyll A, likewise. And then we can go to some of the sort of the macro and micronutrients that we measure using IPP mass spectrometry, and then finally you get to something like iron, where in this case our predictions are, to be honest, pretty bad. But it's also pretty much what we expected, because there's not a lot of iron in these, and it doesn't really absorb strongly. The only way that we probably could sense like a nutrient or an element like iron is if it's a nutrient that if it becomes deficient, then it has symptoms, or if it just generally co-varies strongly with other things. Otherwise, we wouldn't really expect to be able to estimate iron content. So in general, we can summarize the performance of these models with certain traits, like these kind of structural and water-related traits, like equivalent water thickness, cymatic content, LMA, we get really good predictions of. For other traits, like some of our carbon fractions and carbon and nitrogen, we can still get R squared above 0.7. For certain pigments and for lignin, we can also do pretty well. And then at the end, of the tail, we have all these different elements for which our predictions are not quite as good. And I mentioned again that we have transmittance spectra, and using reflectance and transmittance, you can calculate absorptance. For most traits, the models are slightly better when you use transmittance or absorptance spectra than reflectance spectra. I'm not going to show exactly how much, but they're slightly better than with reflectance spectra. You can also ask which wavelengths matter. And here I'm showing the variable importance in prediction or projection metric, which people use as a way to highlight wavelengths that are particularly important for predicting a given trait from PLSR outputs. And so for most traits, so here I'm only showing a few traits, but for most traits, this sort of green and red edge really matter quite a bit. And this is probably because a lot of these traits kind of co-vary with each other to some extent. And so if these traits are kind of central, are important, sorry, those wavelengths are important for predicting a few traits that are sort of central to the network of trait covariance, then it makes sense that other traits might also be, they might also be useful for those other traits. But then there are also, in the near infrared is generally not so important for most traits. And then in the short wave infrared range, we tend to have a few features that tend to stick out for multiple traits. But in general, the short wave infrared is more important than the near infrared. And then we can ask, how well do these models generalize to new sorts of settings? So I put together an independent validation data set. This is from a couple foundational data sets in kind of the study of leaf optical properties. So there's LOPEX and Angers, which are kind of freely available data sets. And then there's the Descent data set that was collected by a student who was in Etienne's lab. But these are all with different spectrometers than the core CABO data sets. And the measurement protocols for traits for LOPEX and Angers at least are fairly different. So I wanted to see, and like they're also sampling different kinds of vegetation. So I wanted to see, do our models kind of transfer to new settings? And again, for certain traits, they seem to transfer quite well. Like leaf dry matter content, it's this model seems to predict things with relatively low bias. Surprisingly, well, I shouldn't say surprisingly, but then for other traits like nitrogen content, again, it seems like we can kind of track the one-to-one line, but there's a fair bit of noise here. For a trait like equivalent water thickness, it seems like there's a strong correlation between our predictions and our measurements, but there's also a considerable amount of bias. So these lines are not at all aligned to the one-to-one line. In each case, the prediction is greater than the measured traits. And then for certain traits, like this is phosphorus as an example, and the predictions are really not very good at all. Some of them are negative. So in cases where our predictions are biased, you might be able to get away with. You might be able to develop a correction factor by measuring a certain number of your samples, and then apply that correction factor to predictions from the remainder of your samples or something like that. But if the predictions are just bad, then you can't really do that. I also looked at how well models transferred among different functional groups, and so training models on all functional groups, functional groups except for one of them, and then testing the model on the one functional group that was left out. So in this case, if you can train a model on conifers, broadleaf trees, forbs, and graminoids, and then test it out on shrubs, and then iterate that with each functional group left out in turn. So the next time, graminoids might be left out, and the rest might be used to train the model and then applied to graminoids. So I'll just show one example of how this might look, and this is a example of where the models are pretty successful. So, well, I guess sort of. I mean, where there's like I think an interesting feature to pull out. So this is for conifers, and when so the model was trained on all the groups except conifers and then applied to the conifer to predict nitrogen. And one thing that kind of sticks out is that you have these two different groups. The predictions aren't necessarily that great within this group. I was pretty happy that this group was pulled out because that's Larix, the one deciduous conifer in the data set, which has clearly different traits like much lower LMA and higher nitrogen content or concentration than the other conifers. So I was pretty happy that the model, which didn't include any conifers at all, was able to pull out this difference between Larix and the remaining conifers. In general, we can summarize the performance for the internal validation, which is taking a random subset of the full CABO data set, the independent validation, which was the 3 other data sets, and the kind of functional group transferability analyses by kind of plotting all of the kind of R² values on the same plot for 4 different traits. In this case, you can see in which case you see a big drop off between the internal validation, which is kind of the best case scenario for validation, compared to these stronger or harsher tests of how well these models transfer to new settings. And in certain cases, for certain traits, there's some drop off, but the models seem to hold up relatively well, at least in some cases. So these include, LMA, HR matter content, equivalent water thickness, nitrogen, and the pigments. And for the other traits, the carbon and the carbon fractions, there seems to be a pretty steep drop off, in that the internal validation seems to work quite well, and well, maybe not as much for lignin, but for the others, the internal validation seems to work quite well. And then there's the as soon as you apply it to anything outside that internal validation data, or like out from a different kind of data set, the performance is considerably worse. And like based on experiences doing similar things with other data sets for other projects like the herbarium project, it seems like my sense is that models for these traits, might not transfer as well to new kinds of settings in general. Whereas I think in the case of like pigments, these models are not necessarily spectacular, but we have a pretty good mechanistic sense of where pigments absorb, and so it kind of makes sense of that the models that we build will transfer pretty well to new settings, I think, intuitively. And likewise for LMA or equivalent water thickness, we know where water absorbs, so it kind of makes sense that those models will hold up in some sense. So what we've learned, well, we succeeded in building models to predict a wide variety of traits across these different functional groups with varying accuracy, but for some traits at least, and some of the really important traits like LMA or equivalent water thickness, we get really quite good accuracy. And models for certain traits seem to transfer well to new settings. Other traits not so much. So what's next? We're putting some final touches on a manuscript summarizing these results. And so students and PIs whose data I included in this project, you'll have a chance to read and contribute to the manuscript soon. I think CABO's policy is that if your data are in here, then it's like an opt-out. Like you'll automatically be included unless you opt out as an author. If you want the code or models, regardless of whether your data are in here or not, just for any purpose, if you're within CABO, get in touch and I'm happy to share. A big thank you to many CABO people whose work is reflected in this project. And yeah, I'm glad to take questions.